Sartorius is a leading international partner of the biopharmaceutical industry. From discovery to process development to clinical and commercial manufacturing, our portfolio of proven solutions helps our customers achieve faster time to market while saving costs. Together, we're setting the standard in cell and gene therapy. Hey, listeners, thanks for tuning in to episode 92 of Cell and Gene, the podcast. I'm your host, Erin Harris. Thanks for joining me. Welcome. On this episode, you'll hear my conversation with Dr. Lothar Roessig. He's the SVP, Integrated Product Team Lead, Congestive Heart Failure at AskBio. Lothar and I get into the weeds about congestive heart failure, or CHF, and why gene therapies are advantageous for cardiovascular diseases like CHF compared to other therapeutic areas. So here's a little bit of background on CHF. Heart failure occurs when the heart cannot pump blood efficiently enough to meet the body's needs, including providing sufficient oxygen to the organs. And congestive heart failure results in the slowing of the blood flow out of the heart, which causes the blood returning to the heart through the veins to back up. This causes congestion in the body's tissues, and sometimes symptoms can include swelling in the legs and ankles, and fluid collects in the lungs and interferes with breathing. Now, approximately 26 million people worldwide are living with congestive heart failure. So with that background information in mind, listen in to hear Dr. Roessig share insight on the current treatment landscape for CHF and how gene therapy is poised to address the root causes of it by targeting the underlying genetic and molecular mechanisms of the disease rather than just managing the symptoms. All right, here's my conversation with AskBio's Dr. Lothar Roessig. Enjoy. Hi, Lothar. Welcome to Cell and Gene, the podcast. Thanks for your time today.
Hello. Thanks for having me.
Absolutely. So talk to us a little bit about AskBio.
So AskBio is a subsidiary, and we call it a fully owned, independently operated subsidiary of Bayer. Bayer acquired AskBio about 4 years ago, and it's now Bayer's gene therapy platform company. I should mention, when it comes to AskBio, I should mention the name of Jude Samulski. Jude Samulski is a pioneer, an academic, highly reputed pioneer in adeno-associated viral vectors for gene therapy. He actually was instrumental and was leading to many of the developments that ultimately led to the first AAV gene therapies in other indications that meanwhile even got approved. So Jude Samulski is a co-founder of AskBio. Meanwhile, AskBio has grown to a fully integrated clinical stage biopharma. So we are running clinical stage programs, 2 of them in phase 2, farthest advanced, that is one in Parkinson's disease, and the other is what I represent here today, the heart failure program. Another maybe important background of AskBio is that we feel committed not just to stay in monogenic rare disease, that's something we also pursue with our gene therapy developments, but also move on to broaden the disease spectrum and actually try and apply the better and better established AAV gene therapy principles to broader disease such as heart failure or Parkinson, as I mentioned.
Excellent. Thank you so much for that explanation of AskBio. And as you said, for the purpose of today's discussion, we're going to talk about congestive heart failure. And so I would like for you to kind of level set the conversation for us by briefly explaining congestive heart failure, or CHF. And in addition to that, tell us a little bit about who CHF typically affects.
Mm-hmm. Yes. So just on my background, I'm a cardiologist myself. I have been working 10 years in clinical practice in Frankfurt at the University Hospital. And I've seen these patients suffering from severe symptoms. Typically, shortness of breath is one of the key problems. There are water accumulations in the body, including the lungs. So what patients suffer from are dyspnea, that means shortness of breath, and they are fairly limited in their physical capacity. And those patients we are particularly interested in are those with more severe symptoms. And unfortunately, despite all the achievements, all the more recent new pharmacologic and device treatments, patients still often reach this more severe stage, and that means they would even start to get short of breath when just climbing one flight of stairs is one example, or walk a block is already difficult. Some patients may report that even getting mail out of their letterbox is something they shy away from, and so this is really a limitation in their daily activities of life. The next thing that is important, and I memorize from my practice, when you try to treat patients perfectly, you discharge them from the hospital. It's just often a couple of months later, you may have your night shift in the emergency room, and the same patient comes again with water in the lungs, with an acute emergency brought to the emergency room for shortness of breath and the need to treat what we call decompensation of heart failure. So these hospitalizations are not only a burden to patients, they are a burden to the socio-economic health systems. They are among the most frequent reasons for patients to come to the emergency room. And even beyond that, they are also associated with high mortality rates. So this is a deadly disease. This is still not much improving despite all the therapies. We have actually seen in the past 10 years still rising rates of mortality from heart failure. So it's a huge unmet medical need. It's a huge disease burden, and there's still a lot to be done on top of available therapies.
Goodness. Okay. Yeah. Thank you for that explanation. And is it, does it typically affect, what age group does it typically affect?
In theory, it could affect any age group, but there is a more frequent incidence and prevalence in higher aged groups. So many patients are in the 60s and 70s. The relative proportion of patients rises. The overall prevalence of heart failure in the US currently is close to 7 million. And it's projected to increase further by 2030. We expect to have 8 million. In 2050, we expect 11 million. So it's an epidemic. It's called an epidemic, and it's still on the rise. Again, despite really good advances in treatment, this is more and more a predominant health problem.
Sure. Okay. And now before we get into certainly AB1002, I want to talk a little bit more broadly about the biggest challenges in developing gene therapies for cardiovascular diseases like CHF compared to other therapeutic areas. So what would you say are those biggest challenges in developing a gene therapy for cardiovascular diseases?
Mm-hmm. Yeah, I may add here that about a year ago, I moved to AskBio in my current role as the Senior Vice President for the product leadership of the heart failure program. Before that, I was working in small molecules, also in the cardiorenal field. My group at Bayer was developing vericiguat and Kerendia, and ultimately filing it towards approval. And so I would say there are, of course, also challenges in small molecules, but also learning. So I think we can learn a lot when we now move with our gene therapies towards those broader populations, broader disease like heart failure. We can learn a lot from small molecules, but what needs to be still learned is to better understand for patients and for our sites when we conduct the trials, for the site coordinators, for study nurses, for PIs, what it actually is, what gene therapy means. There are differences among the field of gene therapy, quite a variety. So there would be ex vivo lentiviral, which is totally different from the adeno-associated virus that we are talking about here. So it is important to better explain and better find the right language to approach sites and people involved, of course, including those who would then talk to patients. And this is an area for us of a constant learning, of an ongoing learning. We just come back a few days ago from the American Heart Association scientific sessions, a big meeting, a big conference in the field of heart disease in the US, in Chicago. And it's not just us, it's more and more also other organizations. There was such an increasing reflection of gene therapy in cardiovascular disease, in heart failure, in other forms of cardiovascular disease, that we can really see that gene therapy is on the rise in cardiovascular. And I'm sure as a community, together with those other research groups, we will be able to take those challenges.
Good. I certainly hope so, given certainly the prevalence of the disease. All right, I want to talk specifically now about mechanism of action, or MOA, of AB1002. So talk to us about what that is, and then how does promoting production of protein inhibitor 1 potentially improve heart function in CHF patients?
Yeah. So let's start with what is available and how this adds, because this would add to current therapy. Current therapy is called quadruple. There are 4 pharmacotherapy agents, 4 substance classes that are considered standard. More or less, roughly every patient with heart failure should be treated with those, and those just tackle compensatory mechanisms. So they would regulate the water homeostasis, the blood volume, if you will, like water pills. They would address the vascular tone, the way the vasculature, the vessels are set. They may regulate the heart rate, which as a counter regulatory mechanism increases. So those are the modes of action of the available therapies. But what we haven't been able to develop yet is something that really strengthens the weak heart. And at this point, I should say that there's among various forms of heart failure, we are talking about here about heart failure with reduced ejection fraction. Reduced ejection fraction means that here the heart is weak, it's dilated, it's enlarged, it's not strong enough to propel the blood as supposed. And so what it needs is to regain strength. We call it contractility. And AB1002 is an agent that aims to as its mode of action, to increase the strength of the contraction. It does so by a molecular mechanism that involves calcium cycling. That is the shift of calcium within the cardiac cells. There is a protein called SERCA. SERCA is an established target. It's a calcium pump that regulates this calcium cycling. With every beat, there is a back and forth of calcium. And SERCA itself was already previously the target. People tried to overexpress SERCA, which is not easy for this protein, but it may actually miss the true target, because the protein amount of SERCA is not altered so much in heart failure. What is altered is the activity. And that's a common finding in molecular pathways, that it's not just the protein amount which matters, but the post-translational modification. That's what we call it, when the activity of a protein is regulated. And in this case, what I1C, our gene product, our insert in our AAV gene therapy does, is upstream of this calcium pump to regulate its activity. As a phosphatase inhibitor, it ultimately leads to a binding protein of SERCA being phosphorylated, and this maintains SERCA active. That's our mode of action, and that goes complementary with available therapies. So it would be a combination partner would be given on top, which is another advantage of AAV gene therapy. It's a single time application. So it's not another daily intake of a pill. You have to imagine, patients do not only take those 4 therapies as pills, they have concomitant medications a lot, diabetes, blood pressure disorders, whatsoever. So oftentimes this is up to more than 10 or even more than 15 pills that they have to take every day. It's called hyperpolypharmacy. And so there is need not only for an additional mechanism, but also for something that can be combined, that does not add more and more pills, but is given at some single time point, and then yields a durable additional effect.
Good. Okay. Now, AB1002 has FDA fast track. It's been designated for FDA fast track designation. So what are the next steps for the program? And then, really, how does it impact your development timeline?
Yeah, so the fast track designation means it's meant by the FDA to facilitate expedited development of new therapies, review of new therapies throughout the development. And FDA designates or provides this designation if there is a therapy being developed to treat serious or life-threatening conditions, and when the potential is demonstrated to address unmet medical needs. So these conditions were fulfilled for FDA to grant the designation of fast track. We are currently in phase 2. As I mentioned initially, the phase 2 so-called GeneFit trial has been initiated in 24 earlier this year in the US and now also in other countries. And we are currently ramping up the recruitment into this trial. It's a trial where we aim at 250 patients in 3 arms. It's a randomized double-blind parallel group trial. And so we are now activating sites and trying to generate data as soon as possible to move on in the development program, and then seek the interaction with FDA based on the data that we are collecting in that trial. You were initially also asking what challenges we encounter. One thing to know about this therapy, our gene therapy AB102, is that it is given intracoronarily. So it is part of, similar to a diagnostic coronary angiography, a very standard procedure in cardiology. It's done a million times in the US every year. Can be done in an ambulatory fashion in a standard setting. And we are currently having one overnight stay, but we hope that this would also be in the future development something that can be done as an ambulatory procedure. Now, the important point for the conduct of this trial is that we are using, because of this local administration, we are able to use lower doses than many other, than the approved other AAV gene therapies. And there's another reason why we can use lower doses. Most of the AAV gene therapies are to a large proportion being sequestered in the liver because of the AAV just having a certain propensity to be turned over in the liver. And we are using a novel capsid. So the vector is designed in a way that it avoids being sequestered in the liver. It's a largely reduced sequestration. And that means more of it, a relatively higher amount, is available for the heart where it belongs, where we want it to be effective. And that means that we can use lower doses, and we can use them to such a low extent that no immunosuppression is needed. And that's another facilitation of our trial conduct. That means that patients do not need to take a complex immunosuppression regimen, but we can do it entirely without immunosuppression. Thus far, in none of our patients that was required. And so we hope that with this rather facilitated lower dose administration, that we are successful to recruit our trial in this large cohort of up to 150 patients and generate the data that would then be for further regulatory interaction.
Wonderful, wonderful. Okay. And Cell and Gene will be following AskBio into 2025 for sure to watch the progress. And that actually is my last question for you. What is the roadmap for AskBio in 2025? What are we really looking at?
So this is, I think, for our heart failure program, all eyes are now on execution of this trial, GeneFit. We are working on this to proceed with the recruitment, and the further dates of the trial are actually web posted on our clinicaltrials.gov recording. The other programs run in parallel. I mentioned that there's another program also in phase 2. And what I also like to mention is that The phase 2 study is not the first study. There is already a data set available from a phase 1, a single arm study with AB1002. Preliminary data had already been published, presented, not published as a manuscript, but presented at previous conferences. And we are also hoping now in 25 that with patients in this phase 1 trial, in the 2 cohorts with 2 doses that were treated in this phase 1 trial, that this could be brought to a presentation at a large cardiology conference in 25. So that's another perspective, another outlook for 25, what we plan.
Oh, excellent. That's so exciting. And I hope that does certainly come to fruition. That's wonderful. All right. That's all she wrote for this episode of Cell and Gene, the podcast, featuring my guest, AskBio's Dr. Lothar Roessig. Lothar, thank you so much for your time, your insight. This was a great, great discussion, and I'm so happy you took your time to talk with me today.
Thank you, Erin, for having me. It was a pleasure.
Absolutely. All right, listeners, thanks for tuning in, and we'll see you next time. Talk soon.